Before we dive into our the topics we're covering in class this semester, we probably need to stop and ask ourselves a really basic question. What exactly is biology? So I know I said it in the first video, and you probably heard it over and over again elsewhere. Biology is the study of life. And when we stop and really think about what that means that covers a lot of ground. It covers everything from the ecosystems around us to the microscopic molecules within our own bodies and the reactions that power life itself. We're in this sort of introductory class, we can't go into incredible detail on all of these, but hopefully by the end of this semester, you'll come to a better understanding of these different scales of life and how they interact together in creating all the life we see around us. Biology includes multiple other fields within it, and these are specialized in looking at very particular aspects of the study of life. This can include things such as genetics, ecology, biochemistry, physiology, anatomy, evolutionary biology, nutrition, most of medicine, and on from there. Biology is an incredibly diverse umbrella under which all of these different topics are just getting at particular aspects of what it actually takes to be alive. There is overlap then with biology as well with many other fields. This includes things like geology. You particularly see this when we go to study prehistoric life. Geography, where things are on the planet and how the cycles of non-living environmental factors influence the life on it, is an important part of both biology and geography. Psychology, of course, there's an overlap between neuroscience and psychology that's hard to disentangle. Anthropology, trying to figure out a lot of aspects that shape human cultures and development. All of these have a lot of overlap with biology. And so trying to draw a boundary around what is and isn't biology is sometimes a little bit tricky. This is going to be an emerging theme that we're going to be grappling with throughout the semester. Biology can rarely easily be confined to a single category, and everything within it is the same story. To study biology is to become comfortable with uncertainty, with blurry edges and exceptions to the rules we try and put in place. As we go through this course, one of the things I would like you to try to remember is that whenever we describe something in simple terms as, well, this is how it happens in biology, we're describing a general rule to which there are almost certainly exceptions. Rather than being afraid of those exceptions, part of what makes biology so amazing to study is embracing those exceptions and understanding how they form part of this patchwork of the diversity of life. So now we've talked a little bit about what biology is. Now we're going to move into the process by which we actually use science to try to understand things about life and more broadly the natural world. Now, I want to say before we start out, I'm going to be in this portion of the video mainly talking about the scientific method, which is the conceptual model of how we go about understanding the world around us that's been in use for, say, the past 500 or so years, primarily in Western Europe and spreading from there. However, it's really important to note that humans have always been inquisitive creatures. We want to understand what's going on in the world around us. And so throughout time and history, going back before we even had the means to write it down, people had different ways of combining their reasoning and their observations to try to understand what was going on around them. I will, throughout the semester, be bringing in insights that we gained from have gained through these other cultural constructions of knowledge, even though we are primarily focused on this one specific way that happens to be the dominant way in our moment and place and time. Scientific inquiry makes use of two main forms of reasoning. Inductive reasoning is a form of logical thinking that uses a whole bunch of related observations to try to arrive at a general conclusion. For instance, meet Hildegard of Bingen. Hildegard is my corn snake, and you will be seeing quite a lot of her throughout these videos. Imagine that I am trying to understand what sort of enclosure makes Hildegard happiest, and by extension, other corn snakes. I might spend a lot of time watching her to try to determine what parts of her terrarium she's using. Is she using the parts with lots of deep soil where she can burrow? Is she using her hide? Is she spending a lot of time tangled up in the foliage that I have provided for her, or the various logs and pieces of driftwood? From all these different observations, I might conclude that, well, she seems to spend most of her time burrowing, and perhaps that's just what corn snakes like to do. And should I ever get more corn snakes, or make her cage larger, then maybe what I should do is maximize that in the design. Now, this is a really natural form of reasoning, and this is one that you probably, as I'm describing it, realize that you have used a lot in your daily life. It's not the only form of logical thinking, though. We can go in the reverse direction. This is called deductive reasoning. In deductive reasoning, instead of starting from a collection of facts, you start from a general principle and then work backwards to make predictions. In this form, we'd say that, well, I know that from what I understand of corn snakes, that they are from the southeastern US. They like a particular type of leaf litter. They live in particular habitat types. And from this, I would make some very specific predictions that she would like, certain types of plants or certain textures of substrate in her terrarium. And I would then be able to go about testing these. Sometimes it's said that, well, some types of science use only the inductive reasoning and others only use this deductive reasoning. And this isn't quite accurate. These two forms of reasoning fit together. Observations lead to general hypotheses or explanations, and then we make predictions that we can explicitly test. Both of these types of reasoning then play into two different main streams of scientific inquiry. However, these two pathways have a lot of overlap, and like the two forms of reasoning tend to be iterative and build on each other, these two types of scientific inquiry then in turn inform and build on each other. Descriptive science aims to observe and discover new things about the world. For instance, this is where we go and we look and try to figure out what types of particles make up the universe. Or we go out and we survey all the different animals that are in a specific patch of forest. Or we notice something about the world and just go in and try to make as detailed of a record as possible of whatever it is we're trying to observe. Hypothesis-based science is based on starting with a hypothesis, usually derived from such observations, and developing ways to explicitly test this. Now, in all honesty, a lot of biologists don't necessarily do a lot of hypothesis-based science in the way that you might be imagining based on your previous examples of it, When you study evolutionary biology, for instance, it's very hard to go back in time and set up a controlled experiment to try to understand evolutionary outcomes. Although, if any of you ever do get your hands on a time machine, let me know, because I have some research questions. So what steps actually make up the scientific method? Let's start at the very first, which sometimes you wouldn't even necessarily think of as part of the method, but it's in fact fundamental to science in general. It's making an observation, noticing a pattern, noticing something interesting, some anomaly, a weird frog you haven't seen before, a change in when butterflies show up from when you're used to. Any of those things. These things are the very foundation of doing science, because without them, what are we even asking questions about? Noticing a pattern is one thing, but if we want to actually understand what's driving it, this is where we get into setting up how to test it. And to do that, the first thing we need to come up with is a hypothesis. A hypothesis is a causal explanation that, okay, the butterflies are showing up earlier because the temperature is getting warmer. This is something that is testable and falsifiable. This means that you can go and look and say, well, the temperature actually wasn't warmer this year. Or you can go and look at you can make observations that would disprove your hypothesis. This is very important because it can be easy sometimes to notice a pattern and have it fit with a causal explanation, but that could be because of any number of factors. It could be that there's some third factor that is controlling both. But so what's important and what we're actually trying to do when we do science is to disprove a hypothesis, because that's much easier to do than to definitively prove something. How do we do this then? From this hypothesis, we make predictions about what we would observe if the hypothesis is true. This is where we get into the if-then statement. If a warmer temperature means that butterflies emerge earlier, then if I look through, say, a bunch of natural history records that somebody took, I'll see that the years that there were warmer temperatures, there are earlier butterflies. And on the years with colder temperatures, they will show up later. Sometimes the if-then statement in itself is called the hypothesis. Some of these steps are so fundamentally intertwined that it can be hard to disentangle that. But it's really important to understand what the importance of this prediction is. This means that when you go forward with testing it, that you know what to look for. If you were to just go in and be like, okay, I'm just going to look, you can certainly learn things from that. But it's a lot harder to analyze and put into perspective and draw conclusions from your data if you don't have these predictions going in. So once we have that hypothesis and the predictions that we've made from it, we can actually test it. Now, what this looks like can vary widely on the discipline that you're doing. If, for instance, you're doing something that's looking at changes over time, it can be rather hard to do that in terms of setting up a controlled experiment. So the test can simply be going back and comparing previous year's records and trying to draw conclusions from that. This can be also instead where you are doing some manipulations experimentally to directly test this. Both of these are equally valid forms of science. one important thing is that whenever you're testing it, you need to do something that is comparing against the experimental condition. Let's say you wanted to test your hypothesis about butterflies and temperature. Okay, well, you could do that by looking at historic records, or let's say you want to get a little bit more hands-on. You set up several vivariums and put caterpillars in them, and then you set the temperatures to certain levels and record how long it takes for them to pupate and emerge as butterflies. Okay, great. But if we want to know from there, well, is it actually the higher temperatures causing it? You need to not just run that experiment at the higher temperature. You need to run it at a base level. This is called our control. And an important thing is that in general, we try to hold every other variable static and the same between our different test units. But the control is very specifically where everything is kind of the baseline. And we draw conclusions about whether our hypothesis is valid based on comparing our experimental conditions to that control group. Once we've done that, we can actually evaluate what we're doing. This is where this can look a lot of different ways. Sometimes it's rather simple. Sometimes it can be very complicated. But these different methods are getting at the same question. Is the hypothesis supported or not? Did we observe those things that we predicted, or did we not? This is where the pathways start to diverge a bit more. So, first, if your hypothesis is supported, great. You're not done though. Science is not something that just is one and done, happens in a vacuum. For this to actually really be usable and accessible, you need to share it with others. Now, for those of us who do science as our main thing, our main job, this usually means we're publishing papers that describe our experiments and our results. It means presenting at groups of other scientists, all those sorts of things. What this looks like for those who aren't doing academic science, it can be a lot more variable. But the important thing is that in some way you're writing down everything that you did, everything that you observed, and your conclusions, and you're somehow making it available to other people. In terms of if you run an experiment in a lab for a class, for instance, this step is usually you turning in your assignment based on it. But it can range from that simple act to publishing in a prestigious journal that's read by thousands of scientists. But what happens if maybe our hypothesis is not supported? So, first, obviously, it's great to go back and say, okay, did everything work the way that we thought it did? But sometimes, a lot of times even, it's just simply that your hypothesis isn't correct. It doesn't accurately describe what's going on in this system. So, in that case, we go back to our hypothesis stage. We know a little bit more now about this system, about this question, about this pattern that we observed. Now we can go back and we can say, well, if it's not this, let's try the next thing that it might be. Maybe it's not, let's go back to our butterflies. Maybe it's not temperature. Maybe it's, let's say that we're living someplace where there are, shall we say, more noticeable seasons. Let's say that it actually, if it's not temperature, maybe it's when the snow melts. We can then reformulate a hypothesis based on based on this, and go through the process again. Each time we go through this, we're building on what we've already done. And even if our hypothesis is validated, if we do find that it's supported, we make predictions that we then observe happening. Even that usually is not the end, because science, a lot of times, most of the time, more questions will be raised just by the process of testing this. And from those observations, the process begins again. So, in closing, the foundations of the scientific method are types of logic and reasoning and things that all of us do really without thinking about it in most cases. Understanding, though, what exactly is going on, the actual processes going on in that logic and in these steps of testing our predictions. This is where the understanding what those look like in practice is a really important part of understanding and trying to learn more about the world around us. It's also a really key skill for critical thinking. And so even if you're not planning on going into science, understanding how the scientific method works will help you better evaluate when people make claims about, say, the fanciest new nutrition supplement, or when people are talking about climate change. Understanding how scientists use this process can help you better interpret this information that's presented to you. And so hopefully, understanding the basics of this will make you better at critically evaluating information and thinking about the world around you.